which we have a bunch of receptors, right? And that we have a bunch of ligand. When endocytosis completes there, now we'll have a bunch of receptors with their ligands. And if these vesicles don't make it into the lysosome, and the receptors and the ligands are not destroyed, what's going to happen to that series of communication steps? They're going to be maintained in time. So what's going to happen effectively is that each of these will continue sending the signal, even though, and here's the key, even though the ligand itself may have been diluted or washed away from the outside of the cell. Right? When we think cell signaling, we immediately think ligand or signal coming to the cell and impacting one of those receptors. But all of this happens in very fluid, very dynamic systems in which if a ligand came in, diffused away from some source, chances are that it's going to diffuse away, right? And unless more signal comes in, that signal will go away at some point. But if the cell endocytoses, the ligand bound to the receptor, the signal can continue, no matter how much signal persists outside of the cell. Yes. So we tend to think of endocytosis mostly as a process for bringing stuff into cells. And because endocytosis is so coupled with lysosomes, we tend to think of endocytosis often as a mechanism by which cells take stuff from the outside, chew it up, and then recycle components. All of that is true. But there are certain circumstances in which endocytosis can be used to regulate cell signaling in a number of ways. One of them is by destroying receptors and therefore attenuating or turning off the signal completely. But then another less intuitive way is endocytose receptors with ligands and perpetuate the signal regardless of what's happening with that ligand outside. Questions or comments, anyone? I'll take that as a no. Okay, so apparently LRP1 is one of these receptors that can through endocytosis end up regulating cell signaling. The authors don't go any further into how the LRP1 might do this in the context of microglia or elsewhere, but they point out to references that apparently discussed it. And then the thing that the authors are mostly interested in is that LRP1 has also been linked to inflammation. And here's where the waters split a little bit. So there is this piece of evidence that they share with us that says that in mice, if you knock out LRP1 and you come up with or somehow obtain LRP1 null macrophages, in these LRP1 knockout mice, macrophages will demonstrate exaggerated response to LPS. Do you guys know what LPS stands for and is? What's that again? Lipopolysaccharide. And what is that?
It's the chain of the.
You're thinking biochemistry, but from a biological point of view, where do we find LPS? Bacteria, right? So LPS is what codes most bacteria. I can't remember if it's all bacteria, but most bacteria. That's.
It's gram positive. I know it's one of the.
So it's probably the gram negative. I'm guessing. I don't know. Whatever. We can find that out. But the thing is that LPS is a very common compound that coats the outside of bacteria. So whenever we'll see the use of LPS here in as part of this work, whenever people are trying to induce some sort of inflammation in systems, they can incubate those systems, be it a whole animal or cells in culture, with LPS, because LPS evolutionarily has become a sign of there are bacteria here, right? And often our immune systems will respond to that. Mostly the non-adaptive immune system, the innate immune system that throughout evolution learned to associate the presence of LPS with danger, right? And an inflammatory response. We don't know if these bacteria are here trying to do us harm or good. Just in case, let's mount an inflammatory response, make sure that we send immune cells to that region, and just in case, right? So if in mice that are lacking LRP1, there is an exaggerated response to LPS, what would you say? Is LRP1 activating or inhibiting inflammation?
Honestly, I would say that it's a mechanism to activate it through the response of the macrophages excreting an inflammatory response. So I wouldn't say it's directly activating it, but it's in part helping that activation.
So if LRP1 is helping with that activation, right, helping the macrophages, and you take it out, would you expect those macrophages to do better in inflammation or worse?
Well, since they can't, I'm assuming they use LRP1 to kind of get that signal
Uh-huh.
from the lipids. So I would say, in terms of inflammation, they would definitely be better. It would cause an inflammatory reaction more rapidly.
Okay, with LRP1.
Yes.
But if we take it out of the mouse, will those macrophages do better or worse at inflammation? Worse. And what did the research show? That LRP1 lacking mice or macrophages did better or worse? Is that what it says? Exaggerated? They actually did better, right? What this phrase is saying, that the macrophages showed an exaggerated response, is that there was more of a response to LPS. There was, in fact, I changed that term. In the paper, they say an exacerbated response to LPS, right? So if the response of LRP1 lacking macrophages was exacerbated, normally is LRP1 helping or inhibiting? Inhibiting. So the conclusion from that observation is that LRP1 inhibits inflammation. When you take LRP1 out, inflammation will go up, will be exacerbated, it will be exaggerated. The classical inhibition of an inhibitor, the whole inhibiting a brake. If you inhibit a brake, now whatever the outcome was will be exacerbated, exaggerated, induced, enhanced, right? Now, it turns out that that is not the only observation linking LRP1 to inflammation. Another piece of evidence is that LRP1 can be proteolytically cleaved to produce S-LRP1, which stands for soluble LRP1. So LRP1 is a protein that is sitting on the membrane, and there can be proteases that, like APP, will chop off a piece, and that piece now becomes soluble and floats away. And we call that S-LRP1. And it turns out that LRP1 can be proteolytically cleaved to produce S-LRP1, which is robustly pro-inflammatory. So within that context, is LRP1 helping or inhibiting inhibition? But you said it first. Activating, yes, yeah, correct. So the idea here is that through SLRP1, if it's true that LRP1 can be cleaved and produce SLRP1, and SLRP1 is pro-inflammatory, then we would say that LRP1 is inducing inflammation, right? Hence, this whole idea of LRP1 can be a friend or a foe of inflammation. In some contexts, apparently, it can inhibit, such that when you take it out from macrophages, now these macrophages are responding better to LPS. But in some contexts, it may be that it induces it, and when you take it out, you may see less inflammation because you're taking away with it this SLRP1 that is pro-inflammatory. Okay? So, the question that they have for this paper is, what is LRP1 doing in microglia? Right? They wanted to know what LRP1 might be doing in microglia. Would it be helping with neuroinflammation or countering neuroinflammation? So my question to you is, if you wanted to know what LRP1 is doing in microglia, what can you do experimentally to try and answer that question? You'd like to know what LRP1 does in microglia? Experimentally, what approach do you take?
Try either an overexpression of LRP1 just to see what the drastic effects are.
Uh-huh.
Having little to no LRP1.
Yeah.
In the normal state, just to see if it has a gradient.
Yeah.
Is it too much? Is it bad, or is it too little?
Exactly. So that's the intuitively, that's the best approach. You want to see what something is doing in a system, either take it out or put more of it in, right? What do we call those kinds of studies if we're taking out something out of a system? Knockout. The friend of knockouts, knockdowns, right? Or we could take an approach of, in some cases, whenever that is possible, pharmacological inhibition, right? So we can use drugs to inhibit the activity of that something. Now, more broadly speaking, all of those are referred to as loss of function studies, right? So we're trying to lose a function from a system and see what's up. The opposite, we put more of it in, we overexpress, we induce with drugs like we did the other day with that LXR receptor. So those are called gain of function studies, right? So loss of function or gain of function. Cool. Perfect. So now you guys mentioned loss of function studies, and we included some knockdowns or knockouts, right? And that's exactly the approach that these people took. They went down the route of knocking out LRP1 and see what would happen in those microglia, whether inflammation would be exacerbated, like the macrophages, or inhibited because now those microglia lost the opportunity to make soluble LRP1. Now, before we go into the data and to the results, I wanted to introduce to you a system that some of you may have heard before, but many of you may not. And that is the Cre-Lox system. So, quick show of hands, had you heard, not that you can explain or anything like that, but had you heard of the Cre-Lox system before? Some of you, right? So that's what I suspected. So don't worry, for those of you who didn't, we'll go through it in gory detail. Remember when we talked about transposons, right? These segments of DNA that have the ability to jump from their current location somewhere else, and in the process generate some sort of genetic heterogeneity, even within individuals that share in all of their cells the exact same DNA sequence and genetic information. These transposons, these mobile DNA elements, can jump from one place to another. We never really discussed the mechanics of that, but it really comes down to the transposon, which is illustrated here as in a very cartoonish manner, has flanking sequences that we call repeated sequences or internal repeats. And somewhere in between those internal repeats or IR sequences, there's going to be a gene that codes for a very unique type of enzyme called a transposase. These transposases, what they do is they have the ability to match up these IR sequences and then do a cutting and pasting such that there is a recombination event between them, right? So imagine that there is these two sequences that are essentially identical to, I said that IR stands for internal repeats. It actually stands for identical repeats. So transposases have the ability to make these IR sequences match up to each other, and then they cut and paste such that now, if we were to follow the sequence from one end, now there's going to be a jump, and then it will continue this way, essentially generating this new DNA molecule that I show at the very bottom, where the transposon is now missing, right? So the transposase has the ability to do that, and then if we follow the other sequence now, this other sequence will loop back and onto itself, and it will become this circular DNA that is the transposon in its transient state. This transposon will move elsewhere and insert itself back into DNA, completing that jump from one location to another. Okay? So these transposases are enzymes that we classify more generally as recombinases and have this ability of cutting and pasting DNA based on some sort of sequence-specific manner. In this case, the transposases coded by transposons can recognize the IR sequences, make them match up to each other, and do these funny recombination events such that the transposon becomes circularized and can move somewhere else. Effectively, what happened though, from the point of view of the sequence where the transposon used to be, is that now there's been a deletion of sequence. Everything that was in between those IR sequences has been taken out, right? And that's something that at some point scientists thought, oh, this could be very useful. This is a natural way that evolution found through transposons to, in a relatively controlled manner, take out sequence from DNA, right? It's sort of like a natural machine for taking certain segments of DNA out of a genome. So once scientists understood the mechanisms by which transposons can cut themselves out, they hacked the system to cut sequences that we are interested in cutting out from genomes. Okay? And the way that looks is if you could flank a target gene of interest that you'd like to at some point take out from a genome, right? With these IR sequences that in this particular case are called LOXP sequences. So in other words, if we could genetically engineer these IR sequences on both sides of a gene that we'd like to delete out of a genome, and then we could offer those LOXP flanked target genes some CRE. CRE is the particular recombinase or transposase that works with these LOXP sites. Then, if we were to put this together, the CRE recombinase would go find those LOXP sites and do the whole matching them up, forming a loop with the sequence in between those LOXP sites and taking that thing out. Normally, under natural conditions, that would be a transposon that does this by itself because it codes the sequence for the CRE recombinase itself, but scientists have hacked this system, natural system, into an artificial one by which we introduce LOXP sites flanking a gene that we would like to delete, and then we offer CRE recombinase separately such that CRE recombinase can go and cut out the gene that we wanted to delete. Yeah.
So what is IR again? What does IR stand
IR stands for identical repeats. Yeah, there's various flavors of them, right? So IR is a very generic term. The one that the CRE LOX system uses, the IRs, are LOXP sites, LOXP sequences. So far, so good? Yeah? Okay. So to understand how we actually use the system, the idea is that I so far mentioned that you would somehow provide this CRE recombinase, right? And the way we do that is by having the sequence that codes for Cre recombinase, shown here, under the control of a given promoter that will drive the expression of Cre wherever we want that Cre to be expressed, right? And then I kept saying very generically target gene, the gene that we'd like to delete. In this case, what these researchers wanted to do is delete the LRP1 gene. So in the context of this paper, what we can do is we can change that target gene for LRP1. So now what they do is, or what they did is take an LRP1 gene that is flanked by LOXP sites, and those in technical jargon are called floxed mice, because now you have flanking. So flanking LOXP sites turns into floxed mice, and there is just a mouse whose LRP1 gene has these LOXP sequences flanking it on both sides. In that mouse, the LRP1 gene functions just fine because the LRP1 gene has these LOXP sequences flanking it, but nothing is cutting it out yet. Right? We need to offer those mice some Cre for that sequence to be excised. Right? And that we do through cell type specific Cre mice. So in these mice, the expression of Cre is under the control of a promoter that is useful to us. And what these researchers chose to use is a mouse that is in which the Cre recombinase is under control of a promoter called LysM promoter that happens to drive expression of things in macrophages, monocytes, some other immune cell type, and microglia. Right? So in these LysM Cre mice, macrophages, monocytes, and microglia will express Cre. Those mice do just fine because Cre is expressed in those mice, but Cre doesn't have anything to go and cut out because there is no genes flanked by LOXP sites. So on the one hand, we have the LRP1 flox mouse. On the other hand, we have another mouse that is the LysM Cre mouse. Now we mate them. And in some of the progeny, if not all of the progeny, depending how lucky you are with your Mendelian genetics, some of the progeny, if not all, will have both. Will have the LysM Cre genetic construct and the floxed LRP1. So in those mice, and only in macrophages, monocytes, and microglia, will there be expression of Cre that will go and excise out the LRP1 gene. Right? Did that make sense?
How did they figure out that LRP1 was a transposon?
No, LRP1 is not a transposon. LRP1 is a mouse gene, right? But through genetic engineering, they introduced the LOXP sites flanking the LRP1 gene. LRP1 is a receptor for diverse ligands that can do endocytosis and regulate cell signaling, blah, blah, blah. So that's what LRP1 is and does in the mouse. All they did was add these LOXP sites on both ends. How they did that, we can have a conversation. It's a long conversation we can have some other time. But for now, just take it from me that through genetic engineering, you can take a regular mouse genome and replace the LRP1 gene with a version of LRP1 gene that is floxed. Okay? Gabi.
Why can't you have a mouse with the floxed LRP1 and the LysM promoter? Both within the same mice instead of having the cross?
Excellent question. Because excellent question. So the question here is, why not make a mouse with both the LysM Cre and the flox LRP1? Why not? Why have two lines and cross them? And the answer is very simple. This modularity allows you to later take that LIS-M-CRE mouse and cross it to a line that has a different FLOX gene. And likewise, you could take these FLOX LRP1 mouse and cross it to a line that has a different tissue-specific CRE. If you kept a line for every possible combination, let's imagine for 1 second, that you have 3 mice, right, with 3 different promoters driving CRE, and 4 mice with 4 different FLOX genes, LRP1 being 1, name your other 3 genes, whatever those genes might be. So if you're following the math, you have 7 lines, right? 3 CRE lines and 4 FLOX lines. Depending on how you cross those guys, you could generate 12 different combinations. And you could be deleting 4 different genes out of 3 different places. So with 7 lines, you accomplish 12 different combinations. What you were saying is we keep a mouse line for every combination. And that it makes it very inefficient because now you would need, instead of 7 lines, you would need 12. And I just ran a very quick and dirty example with 3 on one side and 4 on the other. Imagine hundreds here and hundreds here. So 100 here, 100 different CRE mice and 100 different FLOX mice. So that's 200 mice that you need to keep in a colony. But with those 100 CRE mice and 100 FLOX genes, you could run 10,000 different combinations. So the advantage of these modular systems is that you can mix and match and achieve cell type specific deletion of genes in a much larger number of places for a much larger number of genes with a relatively small number of mouse lines. Did that make sense? It's a good question. Why go through this trouble of crossing and trusting Mendelian genetics and whatnot? Because in the end, it does pay off. Okay. So far, so good, everyone? Yeah? Cool. So that's exactly what these people did. They took LIS-M-CRE mice, crossed them to LRP1 FLOX mice. In the progeny, they isolated microglia, right? And they looked at how that microglia would respond to incubation with LPS and whether or not they would respond with an inflammatory response, as you would expect microglia to do when you incubate them with LPS, right? And what this graph shows is that, let's use the red, the LIS-M-CRE negative, meaning these are mice that it's the progeny that didn't inherit both. Let me put it that way. Right? So these are mice in which the LRP1 gene was not taken out from the genome. In those mice, if you take the microglia and incubate them with LPS, there is a significant increase in the expression of one of these inflammatory molecules called TNF-alpha compared to what you would see if you incubate those microglia with just PBS, saline. Right? So humongous increase in the expression of TNF-alpha when incubated with LPS, the expression of IL-6, which is another inflammatory cytokine, and the expression of IL-1 beta. So that's the white bars. And in every case, there is an increase in the expression of these pro-inflammatory cytokines, as you would expect. But then they took the LIS-M-CRE positive mice, meaning those LIS-M-CRE mice with a FLOX LRP1 gene, so that the LRP1 gene would effectively be taken out. So you can think of the black bars as what you see in LRP1 deficient microglia. And in LRP1 deficient microglia, there was an increase in the expression of TNF-alpha, but there was no significant increase in the expression of either IL-6 or IL-1 beta. So what that was telling them is that when you take LRP1 out of microglia, now you incubate this microglia with LPS and they won't longer produce IL-6 and IL-1. So based on those results, what do you think? Is LRP1 inducing or inhibiting inflammation? No one else? Inducing? Exactly. So within this context, it would be inducing it such that we take it out and now we see less inflammation. Okay? Remember, these guys wanted to see what LRP1 would be doing in these microglia. Cool. So that is in vitro in a way. So this was with microglia that were isolated from these Cre-Lox mice and grown in culture. So they wanted to see if you could see the same thing in vivo, in a whole animal. So what they did is they took again these LISM-Cre negative progeny. So these are mice that, because you don't have the proper combination of Cre and Flox gene, there's nothing taking out from the genome. So for all intents and purposes, it's just a control. And in those mice, what they did is a sham operation. Remember how we talked about sham operations the other day? I didn't think it was going to come so soon. But in a sham operation, they open the mouse, they expose the spinal cord, and just when they're about to injure it, they don't do anything. And then they close the mouse up and let it recover. So it's just a control operation where you do everything that you do to your experimental group except the injury itself. And in a separate group, they actually carried out a PNL, which stands for peripheral nerve lesion. Right? So they injure one of the peripheral nerves. IBA1 is a marker of microglial activation. So when you take a control mouse and you do a sham operation, there is very little activation of microglia. To be expected. You're not causing injury. These mice are controls. No activation of microglia, no inflammation. But if you do injure a peripheral nerve, now microglia becomes activated. They also saw that inflammation is accompanied by a pretty striking increase in the expression of LRP1 itself. These mice have LRP1, right? Because you haven't taken it out with Cre, because these mice were LRP1 Flox negative. So there was no excising LRP1. So these mice do have LRP1, and LRP1 expression increases when you injure a control mouse. And then they look at the co-localization. So essentially, they look at all of the bright green cells that indicate microglia in the top panel, and they superimpose that image to the red cell that indicates high levels of LRP1 expression. And in many cases, they see the combination of green and red that turns out yellow. And it's simply telling them that all those cells that were greener or brighter green, meaning that they were activating microglia, were also expressing a lot of LRP1. So they were very red as well. Very green, very red, very yellow. Okay? What happened in the LISM-Cre positive mice, those in which we did put together Cre and the Flox LRP1 such that LRP1 was taken out? Well, what they say is that there is still activation of microglia, but less than in an otherwise wild-type mouse. There is still activation of LRP1, but less. And the co-localization drops significantly. So now you look at the fewer microglia that were activated, and you see they're green, but they have very little red, if any. Okay? Now, you may be thinking, so hold on a second. I understand how there could be activation of microglia. We've taken LRP1, but LRP1 cannot be the only thing that activates microglia when you injure a nerve. So far so good. The fact that there is still some activation of microglia, that's fine. But how can there be some induction of LRP1 when we took it out? What do you guys think? How can you explain the partial induction of LRP1 expression in a mouse that theoretically you took out the LRP1 gene? But overcompensating with what? You took out the gene. So I understand the overcompensation argument. So there is less LRP1 function, so I'm going to go to the LRP1 gene and make more of it. But you took out the gene. It's a little counterintuitive, right?
You said that the LRP, it's not a transposon, it's not acting as a transposon, the LRP1, after you cut it out. Because if the transposon can integrate itself back into the DNA, couldn't that also be what might have happened?
Excellent. So Ali is saying, well, LRP1 could insert itself back into the DNA. So you think you deleted it, but you didn't. But as you pointed out, LRP1 flux, LRP1 is not a transposon, so it cannot have reinserted itself. So that's more to my point. We did take it out. It didn't go back into the genome. So if we did take it out, if we did delete it from the genome, how come there can be LRP1 induction? Any other thoughts, anyone?
Maybe like other mechanisms that's making LRP, like, I don't know the proper terminology, but there's other mechanisms that's increasing LRP.
The gene is not there, right? Okay, cool. So there may be some, we took out the gene, but who knows? Maybe there is some long-lived mRNA. All right, cool. So I like that. Along those lines?
Maybe taking it out wasn't effective at all. Like, not at all.
That's it. This whole business of Cre recombination and taking out a flock gene is never 100% efficient. And it's never 100% efficient because it depends on the rather stochastic step of Cre finding those LOXP sites in the genome. The mouse genome is a big genome. And we have these, tiny little Cre molecules floating around the nucleus. They may bump into the LOXP sites and do this whole thing. Oh, I'm going to put this together and do the cutting and pasting and take out this flox gene. Or they may miss it. So in fact, in the paper, they tell us that it is estimated that the LIS-M Cre driving deletion of the flox LRP1 is about 85% efficient, meaning that there is approximately about 15% of cells that retain that LRP1 gene. Okay, I really like the idea of the mRNA though. If even if that excision was 100% efficient, you could still have some mRNA that continues to be translated and perhaps translated even more thanks to your compensatory mechanisms that say, hey, I sense less LRP1. We have the mRNA, let's translate it more, please, because I need LRP1. So there could be a combination of both. But in this case, all evidence points to the fact that this process is not 100% efficient. Cool. So, back to reality, back to this whole idea that LRP1 actually helps with inflammation, that it induces inflammation such that when we take it out, inflammation is attenuated, not completely gone, but attenuated. How could that be? So the authors bring us back to the introduction. They say, well, one possible explanation is that LRP1 is shedding this soluble LRP1 through proteolytic cleavage. So there may be enzymes that go shed off some SLRP1. Let's take a look into that. Let's isolate microglia from the spinal cord and study LRP1 shedding in response to CSF1. So before they do that experiment, so far they had been doing their experiments with LPS, right? That thing that says bacteria, bacteria, bacteria. Now they want to try with a different signaling molecule that normally says inflammation, well, not inflammation, but invasion. There is something wrong here, injury, and that is CSF1. So they first wanted to check if CSF1 can induce inflammation, so they take microglia from these mice, and then they incubate them with increasing amounts of CSF1, as shown here. And what the authors say is that there is a dose dependent, meaning the higher the dose of CSF1, the higher the increase in TNF alpha expression and IL-6 expression. Okay? So incubating this microglia in culture with CSF1 works. So with that, they go and see if a CSF1 does anything to LRP1. And what they did is they took the same cells, incubated them with increasing amounts of CSF1, and they looked at the LRP1 beta chain by Western blotting. Turns out that the LRP1 beta chain is the part of LRP1 that stays in the cell after that proteolytic cleavage of the extracellular part of LRP1. So looking at LRP1 beta chain is, in a way, looking at how much LRP1 stays in the cell. And they see that with increasing amounts of CSF1, there is decreasing amounts of remaining LRP1. They do the densitometry as usual, and they show us that cellular LRP1 decreased significantly with increasing doses of CSF1. And conversely, same experiment, increasing amounts of CSF1, now they look at soluble LRP1 or SLRP1, and the higher the dose of CSF1, the more that you find of that soluble LSF1, soluble LRP1 that is floating around. Densitometry shows that numerically, and they can do the statistics on that. In this plot here, they looked at soluble LRP1 present in the culture media of these cells. If you think that LRP1 is being shed off the cells and is now floating around, you should be able to see it in that liquid surrounding the cells, right? We call that liquid surrounding the cells conditioned medium, CM. You'll see it referred to it in the literature as CM, conditioned medium. And it's simply medium that has anything that those cells gave you. So in this case, you would expect that if there is shedding of LRP1, the soluble LRP1 will be present in the conditioned medium. But in the conditioned medium, they also looked at, and they show us that they looked at the cellular LRP1. So why do you think the authors would be showing us that they looked at the cellular LRP1 and they didn't find any? Why would they share with that this experiment? Yes?
So the soluble one is what's going to shut it off of that. So if there's less of that, there's probably more soluble being formed.
And that's a combination, exactly, that's the combination between panels MN and OP. So that balance between the inversely related, totally agree. But why would they actively show us, say, look, I did look at the cellular in the conditioned media and I found none? Why is that important?
Is it to show that CSF1 is the sole cause of that shedding of this LRP?
How does that show you that, though?
Because if there was another thing, then LRP would still be remaining.
Oh, I see where you're going. So what you're saying is, was there anything else that is... Okay, so I will kind of twist your words a little bit because it's along those sides. So what this control block here is doing is basically saying, look, I see a lot of floating LRP1. But when I look at the whole LRP1, I don't see any floating in the conditioned medium. Why do I do this? Why do we do this? Just to show you that the LRP1 that we see floating must be truly shed. Because there is, again, scientists playing devil's advocates with themselves. There is a world in which you find LRP1 floating in the conditioned medium because the cells are sort of bursting into vesicles. They're kind of getting destroyed, getting disintegrated, and releasing into the conditioned media everything they had. If that were the case, then you would expect to see a lot of these LRP1 beta chain as well. So this control down here, subtle, but it's basically saying, look, this LRP1 that we see floating around in the medium is not because the cells are falling apart. It's truly shed LRP1. I don't see any of the whole LRP1 floating, so the cells are intact. I do see some S-LRP1 floating around, and there is a concomitant loss of whole LRP1 from the cells themselves. When you put all of that together, the most reasonable conclusion is that these cells are truly shedding LRP1 as opposed to just disintegrating and releasing everything they have to the conditioned medium. Yeah? Cool. So what have we got so far? And I'm not quite sure we'll make it, so we may have 1 or 2 slides for Wednesday. So far we have that CSF1 causes inflammation. Right? We saw that by the increase in IL-6 and TNF-alpha. We see that CSF1 is causing the shedding of S-LRP1. It is reasonable to hypothesize that CSF1 would cause the shedding of SLRP1, and that is, in turn, what causes inflammation. Remember that we knew from before that SLRP1 is a pro-inflammatory molecule, so why not hypothesize that CSF1 is shedding SLRP1, and that contributes to inflammation? How do you test that? Your hypothesis is that CSF1 is shedding LRP1, and that is contributing to inflammation. How do you test that?
Deactivate CSF1 and then just inject the cells with a bunch of SLRP1.
Cool. So is that a gain of function or a loss of function study? A gain of function study, right? So what Seb is saying is, let's forget CSF1 for now, and let's supply a whole bunch of SLRP1 and see if we can cause inflammation or not. Right? Interesting. That experiment will address the question, can soluble LRP1 cause inflammation? Because what we did is forget about CSF1 for now and simply concentrate on whether or not SLRP1 by itself can cause inflammation. So it's an interesting question, but doesn't address the original hypothesis. The original hypothesis is I start with CSF1, and CSF1 via SLRP1 is causing inflammation. How do we test that? I see you tempted.
I was gonna say you could potentially like look at areas where like cells are experiencing inflammation and test for levels of SLRP1 or other vectors that could cause inflammation.
So basically what you're saying is let's treat with CSF1, and then let's see where the shedding of SLRP1 is happening, right? And whether or not there is inflammation as well. Interesting idea, except that you're still contributing to what is at first simply a correlation. I see a correlation between CSF1 activation and what may be the co-occurrence of shedding of SLRP1 and inflammation. I'm just seeing them happen at the same time, but I'm not establishing a causal relationship between them.
Maybe antagonize CSF1.
Antagonize CSF1. So we take out CSF1 and
I'll take it out, but like introduce like a different gene that like CSF1 is still like present,
Uh-huh.
but its effects are induced,
what effects?
like in the shedding of SLRP1.
There you go. What your idea?
It's along the same, like, would it still work if you did a system with CSF and just normally, and then just take out CSF1 and then see whether or not the amount of SLRP will decrease?
Okay, so two things here, and we're gonna end there, but quickly. So Jacob's idea is we take CSF1, and then somehow we take these out, and then we ask the question, is there still inflammation? Josh, what you are saying is I do CSF1, and then ask the question, is there still SLRP1? Right? With that experiment, you're basically addressing this question. But to address the question of whether SLRP1 is the intermediate between CSF1 and inflammation, take the middleman out, or take the middle gene out. Kill the messenger. We often ask not to kill the messenger, shoot the messenger. Now we want to shoot the messenger down, right? So what we do is we incubate these cells with CSF1, somehow get rid of the shedding of SLRP1, and then ask the question, is there inflammation or not? And we'll have to wait until Wednesday, unless you guys go read the paper.